From the Biological Psychiatry family of journals, this is the Biological Psychiatry Podcast, where we discuss the latest research on psychiatric disorders and their treatments. Thank you for joining us. I'm your host and social media editor, Elvisha Dhamala. Bipolar disorder disrupts mood, cognition, and daily functioning for millions of people worldwide. Decades of research have shown us that certain brain areas look different in people with the condition. But the brain doesn't work as a collection of isolated parts. It works as a network, with white matter pathways acting as the cables or wires that connect brain regions into functional circuits. Understanding how these connections are altered in bipolar disorder has been difficult, largely because most studies have been too small or too variable in their methods to produce reliable answers. A new study from the ENIGMA Bipolar Disorder Working Group tackles this challenge. Joining me to discuss this work is Dr. Leila Nabulsi from the University of Southern California, first author of the paper in Biological Psychiatry. Dr. Nabulsi, welcome to the Biological Psychiatry Podcast.
Thanks so much for having me.
Before we get into the study itself, can you tell us about ENIGMA? What is the ENIGMA Consortium? How does it work? And why is pooling brain imaging data from sites around the world so important for studying psychiatric disorders?
So historically, we know that neuroimaging studies were limited to small samples, inconsistent methods, poor reproducibility. So smaller study would often produce conflicting results that didn't generalize. So that's where ENIGMA kind of started. So ENIGMA stands for Enhancing Neuroimaging Genetics Through Meta-Analysis. It's a global consortium founded and led in part by Professor Paul Thompson at the University of Southern California. And it was created back in 2009 to directly address these limitations of single studies. So the goal of ENIGMA is to coordinate cross-site analysis so that the data collected worldwide is analyzed in a consistent manner, reproducible way through shared analytical protocols, standardized quality control, distributed workflows. Up to today, I believe it consists of 85 working groups in total. Some of them are disorder-focused, so they span across conditions from neurological to neuropsychiatric conditions. But there's also a lot of methods focus groups, and these cover different imaging modalities, genetics, but also emerging AI approaches. So the way ENIGMA operates, it works in a modular way so that methodological advances developed in one area can be rapidly applied across many disorders and data sets. But why does these matter, right? So brain differences in psychiatry are usually subtle. So in smaller studies, there's been a lot of them, obviously, but differences can really be hard to detect. So like I said, some of the study findings are often inconsistent, but only when you bring together thousands of participants across many sites internationally, we can start to improve statistical power, and that's when reliable, reproducible signals can start to emerge. So we think also scale matters for generalizability of findings. So when an effect is consistent across diverse populations, age ranges, clinical settings from all around the world, then you can be a little bit more confident that it reflects the true biology of the disorder.
And in this study, you focused on bipolar disorder. So can you give us an overview of the disorder? What does it look like clinically? How common is it? And why is it so challenging to study and to treat?
Yeah, so bipolar disorder is a chronic, recurrent mood disorder characterized by alternating episodes of mania or hypomania, which is a milder form of mania that doesn't reach the severity of a full manic episode, also depression, and in some cases, psychosis. There are different types of bipolar disorder. There's type 1, that's defined by having at least one full manic episode or depressive episode, which could include psychotic features like hallucination or delusions. And then there's a type 2 bipolar disorder where for diagnosis, we need to see at least one hypomanic episode and one major depressive episode, but there's no full mania. It historically used to be considered a milder form of bipolar disorder, but actually the time individuals spend in these major depressive episodes make it just as serious as the other type. So bipolar disorder affects roughly 1 in 25 adults in the US. So it contributes substantially to the global disability and carries one of the highest suicide rates among psychiatric disorders. Most neuroimaging studies, including ours, are done during euthymia, which is a stable period of mood between episodes. It's easier to scan patients, obviously, but also what's important is that if we scan euthymic patients, then we can capture trait-like features of the disorder and not measure like acute mood state effects. So only roughly 30% of patients respond to the standard first-line agent, lithium. That has been in use since, I believe, the 70s, but most patients require polypharmacy, so the concurrent use of multiple medications to manage all their symptoms, but many remain treatment resistant. So there's no treatment specific to manage the cognitive symptom of bipolar disorder. Many symptoms can even arise after someone takes antidepressants. So this adds to the complexity of management of bipolar disorders.
So, in this study, you're measuring these white matter pathways reconstructed from diffusion MRI. What exactly are these white matter pathways, and what does it mean when they're disrupted?
The brain has two main types of tissues, the gray matter, which contains the cell bodies, and then we have the white matter, which lies beneath and is made up of long nerve fibers wrapped in a fatty insulated sheet that is called myelin. If we think of gray matter as the city and white matter as the motorway connecting them, without motorways, the cities would just exist in isolation, so they wouldn't communicate. So that's what white matter pathways do. They're the brain's communication cables, so they carry electrical signals between regions. And the speed and reliability of this communication depends on how well myelinated these fibers are. So, how do we measure them? White matter pathways obviously can be seen directly with standard brain scans. We operate at the macro scale with MRI, but one of the acquisition protocols of MRI, diffusion MRI, gives us an indirect window, let's say, by tracking how water molecules can move through the brain. In a well-organized fiber bundle, water moves preferentially along the direction of the fibers, like let's say water flowing through a pipe. So the scanner picks up on this signal and produces the images that as researchers can go and investigate. And so from these images, we can use sophisticated cutting-edge algorithms to reconstruct the pathways in the brain. And that's a technique that we call tractography. And we can derive several measures that describe the organization, the density, and microstructural properties of white matter connection. In our study, we focused on two key measures, fractional anisotropy, or FA. This measure reflects how well organized each fiber bundle is. And then we also focus on the number of reconstructed streamlines between regions, and that gives us an estimate of the connection density. And so when we talk about white matter disruptions in bipolar disorder specifically, what we mean is that the organization of these pathways is altered. For example, if we see lower FA, then we would assume there's a less coherent fiber bundle organization, and that could reflect several changes at the microstructural level. What is important is the consequence of this change is that we have slower, less reliable communication between brain systems. And in our study, we found disruption not just at individual tracks, but in the network level organization of connections across the whole brain network.
So, in this study, as you mentioned, you're combining brain scans from 16 different sites, which use different scanners and different protocols. How do you go about harmonizing all of that data to be able to study it together?
Yeah, that's a very important question. That's one of the challenges of ENIGMA, actually. And for researchers who are analyzing and organizing the data, of course, it does take a village. Obviously, different sites use different scanner brands, different protocols, acquisition parameters. All of these can introduce systematic differences that are unrelated to the biology of the disorder that is what we want to study. And so that's what we call noise. So we collected raw scans from all these 16 sites. They sent those scans to us, to this central processing team here at USC. So an important step was to verify that each site's acquisition protocol was suitable for the tractography-based connectome reconstruction that we wanted to do. All data was then processed centrally using standardized pipelines. Scanning site also was explicitly modeled in our statistical analysis to try to separate this between sites variability from the biological signal.
Two concepts that come up throughout your paper and your results are network integration and network segregation. Can you explain what those mean in the context of the brain and why the balance between integration and segregation matters so much?
So network neuroscience emerged in the late 2000s. So it's quite recent. It borrows from graph theory. That's a branch of mathematics they used to study everything from social networks to the internet, but also to study the brain, not an isolated region, but as an integrated system of connections. So these concepts gave rise to the concept of the human connectome. Basically, that's a complete map of how every region in the brain is connected to every other region. So these are complex concepts that we can break down with, I guess, a simple analogy. If we think of the global air traffic network, we have airports that could be our brain regions and flight routes that are the connection between them. So you wouldn't assess that network by looking at each airport in isolation. You'd ask how efficiently passenger can travel between any two cities, whether certain hub airports carry a disproportionate share of traffic maybe, and whether regions cluster connect well to the global system. So the brain works exactly the same way, and network neuroscience is a very cool tool that lets us ask the same questions but about the brain communication. So there are two important concepts in network neuroscience: integration and segregation, like you mentioned. There's been several graph theory studies conducted so far, not just in bipolar disorder, but in other conditions. And these studies of the human connectome in general, also, people with no diagnosed disorder, have shown that the brain has a tendency to organize into two complementary properties, so the segregation and integration, and that the balance between these properties is fundamental to sustain healthy brain function. So, network segregation refers to the brain's tendency to cluster into specialized structural communities. So, regions that work together, group tightly, each dedicated to specific function like emotion, memory, perception. And this clustering is important because it supports the efficiency with which information is processed locally within these communities. And so one would think that when segregation breaks down, the brain loses its ability to process information in a focused, organized way. And then there's network integration. So that process refers to how efficiently the information travel across the whole brain, allowing the specialist communities to rapidly coordinate with one another. And so when integration breaks down, communication becomes less direct and less efficient. Connecting these two properties are also a set of highly interconnected brain regions. These are known as hubs, like our airport hubs. Maybe Atlanta could be one in the US. So these form a structural backbone that support this global neural communication and support higher order cognitive functions. So one would think that damage to this core system creates a vulnerability in the network. And so the brain needs both segregation and integration in balance. And when the balance is disrupted, like we see in bipolar disorder, this is associated with alteration in how the brain supports thoughts, emotion, behavior, and how really each part of the brain communicates with one another.
So, in your study at the whole brain level, you found that individuals with bipolar disorder had lower network density, longer path lengths, lower efficiency, and higher betweenness centrality. What exactly does that mean in terms of integration and segregation, and just how information flows in their brains overall?
Yeah, so all these findings aren't isolated. So they paint a coherent picture of what the brain network that it looks like in bipolar disorder, and it looks like it's less well connected, less efficient at transferring information, more dependent on a small number of critical bottlenecks. So if we think back to the traffic analogy, if you imagine a network where fewer routes exist, then journeys will take longer. Fewer passengers can move efficiently across the system, and everything funnels through just a handful of overloaded hub airport. So that's essentially what we're seeing in bipolar disorder at the level of the whole brain. But so if we want to break down each metric individually in more plain language, we found lower density relative to those individuals who did not who were not diagnosed with the illness. So that means fewer connections are present across the whole brain network. So the wiring is sparser. So some regions that would normally communicate directly are less reliably connected. We found longer characteristic path length in bipolar disorder. So that means on average, it takes more steps to get from any one brain region to any other. So information has to travel via more intermediate regions rather than direct routes. It's like having to take 3 connecting flights instead of 1 direct. So we also found lower global efficiency. That's a direct consequence of the lower density and longer path length. So the brain is less efficient at integrating and distributing information across the whole system simultaneously. And then we found higher betweenness centrality. So that lets us think that there's a small number of hub regions in the brain that are now carrying a disproportionate share of the network traffic. So more journeys are being routed through fewer airports, making these hub critical bottlenecks. Together, these findings do paint a picture of the brain network that is less well connected, less efficient at transferring information across the network overall, more reliant on a small number of critical hubs to maintain communication, so a more fragile architecture overall. So a network that depends heavily on a small number of hubs is also more vulnerable to disruption. So if one of these hubs is affected, we can think the consequences ripple across the whole system.
You also found 2 specific subnetworks that were primarily disrupted in bipolar disorder, one involving the limbic and basal ganglia regions, and another linking default mode salience and fronto-limbic areas. Can you tell us a bit more about those specific networks and circuits overall and what roles they play?
So the first subnetwork that you mentioned is the limbic basal ganglia circuit. So we found altered white matter microstructure, FA, predominantly between and within limbic regions and basal ganglia nodes. The limbic system, we can think of it as the brain's emotional core, and the basal ganglia are central to reward, motivation, goal-directed behavior. So together, this circuit supports how we experience and regulate emotions, how we respond to reward and threat. So the changes we see here makes us think that white matter connection between these regions are altered in bipolar disorder. It could explain for some of the functional deficits we know that characterize the illness. And then the second subnetwork we found was altered connection density, so number of reconstructed streamline, and spanning across the fault mode network nodes. This is the network, let's say, of the inner self. It kind of supports self-referential thinking, autobiographical memory, but also salience network nodes. So that's like the brain's relevance detector and fronto-limbic connections as well. So it's quite like a wide network, linking prefrontal reasoning with the limbic emotional system. So these are 3 important circuits within the subnetwork. And so when the circuit is disrupted, the brain may struggle to assess what is emotionally relevant. So it may affect the ability of an individual to distinguish internal from external states, um, uh, difficulty in recruiting what we call top-down control, so the ability to pause and regulate on emotional responses and then acting on it when emotions escalate. And so these are conclusions, obviously, that would be great to test in the future using detailed behavioral and cognitive measures, but the anatomical location of our findings make us think we can pinpoint towards those cognitive deficits of bipolar disorder. It's interesting to also note that within both of these 2 subnetworks, we found a mix of lower and higher connectivity. So this makes us think that there are both disruption, but also potentially compensatory processes that are happening. So when core pathways are compromised, then the brain recruits alternative routes to maintain communication and possibly to maintain a euthymic state of the illness.
So, along those lines, you also looked at specific clinical features and how they were related. So you found that longer illness duration, later age of onset, and a history of psychosis were associated with greater network disruption. What does that tell us about how the illness might be shaping the brain over time?
One of the most important thing we can do in a study like this, and with the power of ENIGMA and rich phenotypes, is go beyond simply comparing bipolar disorder to controls. So, and we can ask, within the bipolar disorder group, does this network disorganization vary with how severe or how long standing the illness is? If brain network disruption tracks with illness burden, that's perhaps evident that these aren't just pre-existing traits, that they may reflect something that accumulates or evolves within the disorder. So we found in relation to illness duration that the longer someone has lived with bipolar disorder, the more the brain's communication network appears to be affected overall. And regionally, with reduced connectivity specifically between the hippocampus to the amygdala. So 2 regions that we know are central to emotional memory, fear regulation. And this is consistent with the idea of cumulative disease burden, that maybe there are chronic inflammation and changes in the brain tissue happening over time, gradually affecting white matter pathways that keep brain systems connected. And we also found that later illness onset was associated with more pronounced network alteration globally and in specific circuits connecting the cerebellum, the thalamus, fronto-limbic pathways as well, all networks that are involved in emotion regulation. Usually, earlier onset is associated with greater clinical severity. So deficits, more pronounced deficits seen at a later onset of the disorder maybe are just reflecting differences in treatment timing, in the illness recognition, or maybe there are some underlying biological and clinical factor, perhaps genetic or resilience, that allows an individual to tolerate illness burden before symptoms emerge. We also found that individuals with a history of psychosis showed more fragmented and less efficient brain network, fewer connection, longer communication paths, and greater over-reliance in a small number of hub regions. So this is consistent with prior work that shows that psychosis in bipolar disorder is associated with more severe white matter changes. We believe our findings just extend that picture to show that psychosis leaves a detectable mark on the brain's overall communication architecture.
You also looked at manic episodes, which are another characteristic aspect of bipolar disorder, as you mentioned, and you found that they were associated with higher connectivity in a fronto-subcortical circuit. How do you interpret those findings?
We found that a greater number of manic episodes was associated with a higher connectivity between the anterior middle frontal cortex and some subcortical regions just below the thalamus. These are kind of like in the opposite direction from all the other findings in the study. But we believe one possible interpretation is that perhaps mania induces some sort of neuroplasticity in the brain. So having had repeated manic episodes perhaps drives structural reorganization of this fronto-subcortical circuit through the brain's reward and excitatory signaling systems, because those are like the neurotransmitter systems that are passing through this circuit that we found. And so we think of it like if roads get used so frequently, maybe it gets widened. So there's a structural trace of all this repeated activation. We did not record any connectivity change associated with having experienced a greater number of depressive episodes. I suppose this is a reminder that bipolar disorder is not just a single uniform process, that perhaps different aspects of this illness can leave different biological signatures in the brain.
Earlier on, you introduced the idea of polypharmacy, which is that most people with bipolar disorder are taking multiple medications. And in this study, you examined several different medication classes, also using something called the neuroscience based nomenclature. Can you tell us a little bit more about that approach and what you found?
The challenge in studies like ours is separating each drug effects from others and from the illness itself. So within ENIGMA, we had this wide pool of information, clinical information, including medications. And so we decided to tackle this issue by modeling current medication and illness severity together in order to identify those effects specifically and separate those effects from the illness effects. And so we chose to examine medication under two complementary frameworks. So the conventional indication-based categories and then the neuroscience-based nomenclature or NBN. So the conventional categories group drugs by what they're prescribed for. So lithium, antipsychotics, antidepressants, anticonvulsants. It's clinically intuitive, but neurobiologically, it's a bit blunt since drugs within the same category could act on very different mechanisms in the brain. And we're looking at anatomy, so we want to be as precise as we can on effects that we're measuring. So the NBN group drugs instead by what they actually do to the brain. So instead of antidepressant, you'd call it a serotonin reuptake inhibitor. So it's kind of like a shift from what is the drug used for to how does this drug act on the brain. So the most significant findings were actually seen for antidepressants and antipsychotics. So for antidepressants use, particularly, we found that the SSRIs, serotonin reuptake inhibitors, were associated with less efficient communicating brain network, weaker connection between two limbic system regions, specifically the thalamus and the hippocampus. And then we found that a class of antipsychotics acting on multiple neurotransmitter system, was linked to weaker connection, specifically in the orbitofrontal cortex. That's a region involved in emotion and decision making. We believe a take-home message from our medication analysis that treatment exposure is an important factor to consider when studying the biology of any disorder, really, but in particular bipolar disorder. People receiving certain medication may also differ in illness history or symptom severity. So separating the effects of the illness from those of the medication is key in future study. And we do hope that our approach can encourage future studies to take these factors into account.
Interestingly, you didn't find any significant associations with lithium use. What does that tell us?
We found that lithium was actually, the only medication class that showed no significant association with any of our network connectivity measures, while antidepressants, antipsychotic, anticonvulsant, they all left some sort of detectable, let's say, footprint on our network. So one interpretation is that rather than altering the network, lithium may be stabilizing it, preserving structural connectivity closer to its baseline, while other medication perhaps leave a more measurable mark.
So, if we take these findings together, what does that mean for the day-to-day experiences of someone living with bipolar disorder? So, in terms of their actual daily functioning, in emotional regulation, decision-making, or cognitive function?
Our study measures brain structure, so not symptoms directly. What we're saying is that the circuits we found disrupted are the same ones that are implicated in emotion regulation, cognition, reward, by a large body of prior work. And since the vast majority of our participants were euthymic at scanning, we think these findings reflect the persistent trait-level biology of bipolar disorder, what's present in the brain when someone is clinically stable. But specifically, what our findings may mean for different cognitive aspects of this disorder is that, for example, in relation to emotional regulation, the disruption within fronto-limbic circuits is the pathway through, let's say, which the frontal cortex exerts this top-down control over the limbic system. When less well-connected, emotions may escalate more quickly, be harder to bring back down. The salience network disruption may further contribute to some stimuli that don't carry an emotional valence felt disproportionately intense or threatening. The reduced network efficiency and hub over-reliance offer another structural basis, I think, for the persistent cognitive difficulties we see in bipolar disorder, even during euthymia. So sustained attention, working memory, cognitive flexibility, these may all be affected. And a disrupted default mode connectivity, it could be consistent with difficulty disengaging from rumination. We also have basal ganglia disruption. Those are central to reward processing, motivation when their connections are altered, their ability to assess risk and reward, feel motivated, sustain goal-directed behavior. Those could also be all compromised. Big picture is if we can understand where symptoms map onto the brain and where existing interventions map onto the same circuit, then we can begin to think about how to target them more precisely. And I think this is the long-term promise of this kind of work.
If there's one key message you'd want clinicians, researchers, people affected by bipolar disorder, and their families and friends to take away from this work, what would that be?
Bipolar disorder is a disorder of brain communication. And being able to understand that at a scale across the whole network and the full course of the illness through collaborative efforts like ENIGMA is how we will eventually move psychiatry towards more objective, biologically informed decision making and treatment that are truly matched and can really make a difference to the individual.
That's all for today's episode. Thank you to Dr. Nabulsi for joining us to share her work on white matter disruptions in bipolar disorder. For our listeners, you can find the full paper in Biological Psychiatry, and we'll include the link in our episode description. To learn more about our journals, please visit biologicalpsychiatryjournal.com. You can find the Biological Psychiatry Podcast wherever you get your podcasts. Subscribe now so you don't miss our next episode, and follow us on our social media channels for highlights and updates. Thanks for listening. This podcast is for informational purposes only and does not constitute medical or professional advice. The views expressed are those of the participants and do not necessarily reflect those of the Society of Biological Psychiatry, its family of journals, or its editors. All rights reserved, including those for text and data mining or use in AI systems.